The goal of HubMap is to create a reference atlas of the healthy human body at single-cell resolution. To achieve that goal, researchers must be able to name and map the trillions of cells and the many distinct cell types that make up the human body. Different cell types perform different functions. While some cell types appear throughout the body, some only exist in certain organs. Authoring and organizing information about all those different cells is the role of HubMap's Anatomical Structures and Cell Types plus Biomarkers, or ASCT plus B, tables effort.
So the ASCT plus B tables are basically a scientific organizational framework where we take anatomical structure, AS, CT, cell types, and biomarkers, and organize this information into an ontologically relevant framework where many different researchers and many different data managers from different backgrounds can collectively see, align, understand, and then use information about biological taxonomies across the whole body.
Take, for example, the renal corpuscle. It is part of the kidney and has different substructures, such as the glomerulus, that are made of different cells. This is the ASCT plus B table for the kidney. Let's zoom in to the renal corpuscle, which includes the glomerular tuft and other substructures. The table also indicates the cell types that make up each anatomical structure. Plus, the table lists the biomarkers that can be used to identify cell types. We see that the glomerular tuft contains 2 cell types, which are commonly identified by specific genetic biomarkers. The ASCT tables are flattened to make them machine-readable so they can be visualized for exploration.
ASCT plus B tables are complex. In fact, each table represents a pathognomy tree and 2 bimodal networks. To help experts author these tables and reviewers review them, we implemented the ASCT plus B reporter visualization. Using that visualization, you can interactively explore not only the anatomical structures hierarchy, but you can also select, for instance, an anatomical structure and see what cell types are associated with that structure. You can also select a cell type to see what biomarkers are typically used to identify that cell type. In addition, you can use the ASCT plus B reporter to see which anatomical structures have that particular cell type. You can do this not just for one organ, but also for now 10 organs and soon more. For example, you can search for a specific cell type across all 10 organs and start to understand in what organs that particular cell type exists and which biomarkers are commonly used to identify that cell type.
Tables are authored by a team of subject matter experts. Anatomists, pathologists, and surgeons decide on major anatomical structures and their part-of relationships. Single-cell experts work towards standard definitions and characterizations of cell types and their biomarkers. All work closely together to add AS, CT, and CTB linkages based on the rich scientific data that is becoming available. Draft tables are then sent to a second set of experts who review them for scientific rigor, coverage, and quality. Throughout this process, care is taken to ensure that all ASCT plus B tables are in close alignment with existing ontologies like Uberon or CL to support effective collaboration and data sharing. Discrepancies with existing ontologies, for instance, the addition of new terms or term definitions, are resolved in close collaboration with ontology curators.
So one of the reasons why these ASCT plus B tables are important for biomedical research is because there really aren't enough standards in the field for biomedical researchers to use and align and share information on. So this kind of table provides sort of like a Rosetta Stone for people to share what's known about their individual fields using collective ontologies and languages that then can be refined or updated in a sort of common framework for the rest of the field?
Well, I mentioned before how standardization is essential to understanding the information that's being generated and collected by the research community. This standardization makes possible the development of reference spatial systems that provide a means for organizing community data and information by spatially indexing this knowledge with respect to a coordinate system defined over the human body part of interest. These anatomical coordinate systems, or what are sometimes called common coordinate frameworks, serve as atlases for the human body and allow us to pinpoint the location of, for example, where tissue samples are taken, where in vivo biomarker measurements were observed, and so on.
ASCT plus B tables are used to design HubMap's Common Coordinate Framework, or CCF. Specifically, they're used to develop anatomical structure partonomies and cell type typologies in support of semantic search. The tables are also used to design semantically annotated 3D reference organs in support of spatial registration, mapping, and exploration. As new cell types and biomarkers are discovered, the tables will need to be expanded.
The ASCT plus B tables are essential in defining our GPS systems for the human body because they provide a unified place name for every location or position in the body.
Currently, ASCT plus B tables exist for the brain, colon, heart, kidney, and spleen. Tables for other organs are still under development. If you are interested in contributing new tables or reviewing existing ones, please fill out this online registration form, and we will send you information and meeting invites. Or simply contact us at info.ccf@indiana.edu.